Hello dear students. So in today's class, let us discuss about the glycolysis. So that is anaerobic carbohydrate metabolism. So glycolysis is nothing but, so it is the sequence of steps that involves the conversion of glucose into pyruvate with the release of ATP molecules. So glycolysis is the sequence of steps, so which involves the conversion of glucose into the pyruvate and with the release of the ATP molecules. So that is called as the glycolysis. So for this glycolysis process, so the detailed pathway was given by three scientists. So that is Emden, Meyer, and Parnas. So that's why the pathway for the glycolysis process is generally called as the EMP pathway. So that is EMP pathway of glycolysis. So according to this EMP pathway, so the glycolysis occurs in two stages. So that is, the first phase is the preparative phase and the second stage is the payoff phase. So these two stages contains a total number of 10 reactions. So let us see what is preparative phase and payoff phase. So see here, in the preparative phase, so the glucose molecule will break down into glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this process, so that is the conversion of glucose into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, so that requires energy. So therefore here what happens in the preparative phase, so the glucose molecule will consume the two molecules of ATP. So by consuming two molecules of ATP, the glucose will break down into the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this is preparative phase. So in preparative phase, the glucose molecule consumes two molecules of ATP and breakdowns into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this is the first stage in the glycolysis, preparative phase. So in the payoff phase, so the triose phosphate is converted into the pyruvate. Okay, this is the second stage in the glycolysis. So the triose phosphate, okay, so that is converted into pyruvate. So each molecule of glucose can produce two triose phosphate. Okay, so each glucose will produce two molecules of triose phosphate. So two molecules of pyruvate will be formed. So when two molecules of triose phosphate, so they undergo payoff phase, so they will produce two molecules of pyruvate. Okay, so during the formation of each pyruvate, two ATPs are produced. Okay, so thus in a glycolysis process, okay, so the two ATPs in glycolysis, two ATPs per molecules of glucose is produced. Okay, for each molecule of glucose, two ATPs are produced because each glucose molecule will form two triose phosphates. Each, that is two triose phosphates, will form two molecules of pyruvate. So that this two molecules of pyruvate will form two molecules of ATP. So therefore, in the glycolysis process, per molecule of glucose, two ATPs are produced. For one molecule of glucose, two ATPs are produced. So next let us see the EMP pathway. So we know that the glycolysis process involves two stages, preparative phase and a payoff phase. So these two phases will contain a total number of 10 reactions. Okay. So let us see the 10 reactions. Let us discuss about those 10 reactions. So the first step in the EMP pathway, that is glycolysis of glucose, the step 1. So in step 1 what happens? So the glucose molecule, consumes 1 ATP in presence of hexokinase enzyme to form D-glucose 6-phosphate. So that means this glucose molecule gets phosphorylated by consuming one molecule of ATP in presence of the enzyme called as hexokinase. Okay, so glucose in the step 1, glucose gets phosphorylated into glucose 6 phosphate. Okay, so here in the step 1, one molecule of ATP is consumed. Okay, so next let us see the step 2. So here you can see in step 2, the glucose 6 phosphate, so which is formed in the step 1, so is converted into fructose 6 phosphate. Okay, so in presence of glucose phosphate isomerase enzyme. Okay, so glucose 6 phosphate is converted into fructose 6 phosphate in the second step. So here you can see, so in the second step, so the glucose 6 phosphate in presence of glucose phosphate isomerase enzyme converts into fructose 6 phosphate. Okay, so that is step number 2. So in step 3, the fructose 6 phosphate which is formed in the second step is converted into fructose 1,6 diphosphate. Fructose 1,6. So 1,6 diphosphate in presence of the enzyme phosphofructokinase. Okay, so this is fructose 6 phosphate. Okay, it has only one phosphate group at the 6th carbon atom. So further it gets phosphorylated. Okay, so fructose 6 phosphate gets phosphorylated in presence of phosphofructokinase enzyme to form a diphosphate. Fructose, it's a monophosphate. It's converted after phosphorylation, it gets converted into a diphosphate. So fructose 1,6 diphosphate is formed. Okay, this is step number 3. So in step 4 what happens? So the 6 carbon fructose 1,6 diphosphate, so which is formed in the step 3. Okay, so is breakdown into the 3 carbon sugar phosphate. Okay, so that is the fructose 1,6 diphosphate in presence of the enzyme called as aldolase. So this 6 membered fructose 1,6 diphosphate is breaks down into 3 carbon sugar phosphates. So the first one is dihydroxyacetone phosphate, which is called as DHAP, dihydroxyacetone phosphate, and one more is the glyceraldehyde 3 phosphate. Okay, so the fructose 1,6 diphosphate breaks down into a 3 carbon sugar phosphate. So next let us see the step number 5. So in step 5, so the dihydroxyacetone phosphate, so that is which is formed in the step 4, the 3 carbon containing sugar. So this dihydroxyacetone phosphate DHAP, so isomerizes to glyceraldehyde 3 phosphate. Okay, so this DHAP, so that is dihydroxyacetone phosphate, which isomerizes in presence of the enzyme called as the triose phosphate isomerase. Okay, so this dihydroxyacetone phosphate DHAP is isomerizes into the glyceraldehyde 3-phosphate. Okay. So next let us see the step number 6. So in step 6, so the glyceraldehyde 3-phosphate, which is formed in the step number 5, undergoes oxidation to give 1,3 diphosphoglycerate. So in presence of an enzyme called as the glyceraldehyde 3-phosphate 3-hydrogenase enzyme. So this glyceraldehyde undergoes oxidation in presence of this NAD plus and inorganic phosphate to form 1,3 diphosphoglycerate. So that is oxidation of this glyceraldehyde 3-phosphate into 1,3 diphosphoglycerate. So next let us move on to the step number. So in step 7, this diphosphoglycerate, so which contains the 2-phosphate group, so it's generally it's a high energy molecule. Okay, so therefore in step 7 what happens, so this high energy 1,3 diphosphoglycerate transfers the phosphate group to the ADP. So adenosine diphosphate. So one phosphate group. So here you have the 1,3 diphosphoglycerate has 2 phosphate groups. So one phosphate group is transferred to the ADP. So this ADP is converted into ADP. Diphosphate, adenosine diphosphate will converted into adenosine triphosphate, that is ATP. So in presence of the enzyme called as the phosphoglycerate kinase enzyme. Okay, so this is the 1,3 diphosphoglycerate is a high energy molecule because of the presence of 2 phosphate groups. So therefore this high energy 1,3 diphosphoglycerate is transfer one of its phosphate groups to the ADP, so that ADP it gets converted into ATP, and this diphosphoglycerate is converted into monophosphoglycerate, that is 3-phosphoglycerate. So next let us move on to the step number 8. So in step number 8, the 3-phosphoglycerate is isomerizes. So isomerizes into 2-phosphoglycerate in presence of an enzyme called as the phosphoglycerate mutase. So the monophosphoglycerate, so that is 3-phosphoglycerate, which is formed in the step number 7, is undergo isomerizes, or which isomerizes into 2-phosphoglycerate in presence of the enzyme called as the phosphoglycerate mutase. Okay, so next step, so in step number 9, so the phosphoglycerate, the 2-phosphoglycerate which is formed in the step 8 is dehydrates, which undergoes dehydration to give phosphoenol pyruvate. So which undergoes the dehydration. So which dehydrates the 3-phosphoglycerate dehydrates in presence of an enzyme called as enolase to form phospho, so which converts into the phosphoenol pyruvate. So next let us see the last. So the last step in the glycolysis is the conversion of this phosphoenol pyruvate into pyruvate in presence of an enzyme called as the pyruvate kinase. So this is the phosphoenol pyruvate. So in presence of the enzyme pyruvate kinase, so the phosphoenol pyruvate is converted into pyruvate and the ADP. So the phosphate group of the phosphoenol pyruvate is transferred into the ADP. So that ADP is converted into ATP and the phosphoenol converted into pyruvate. So this is the last step in the glycolysis process. So here you have the EMP pathway, the representation of EMP pathway. So the glucose, it consumes one molecule of ATP and gets converted into glucose 6 phosphate. So that is the phosphorylation of glucose in presence of hexokinase enzyme to form glucose 6 phosphate, where one molecule of ATP is consumed. So this glucose 6 phosphate in presence of phosphoglucoisomerase enzyme, so is isomerized into fructose 6 phosphate. Okay, so this fructose 6 phosphate consumes an ATP molecule to form the monophosphate. Fructose monophosphate is converted into fructose diphosphate. So that is fructose 1,6 diphosphate in the presence of the enzyme phosphofructokinase. So this fructose 1,6 diphosphate in presence of the enzyme aldolase is converted into, is breakdown into the 6-membered fructose is breaks down into, so 3 carbon sugar. So that is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Okay. So this dihydroxyacetone phosphate DHAP in presence of phosphate triose isomerase, so it isomerizes into glyceraldehyde 3 phosphate. Okay, so this glyceraldehyde 3 phosphate in presence of glyceraldehyde 3 phosphate hydrogenase enzyme is converted into 1,3 diphosphoglycerate. Okay, it's oxidizes into 1,3 diphosphoglycerate. So this 1,3 diphosphoglycerate, so is converted into 3 phosphoglycerate and the ADP are converted into ATP molecules. So this 3 phosphoglycerate, 3 phosphoglycerate in presence of phosphoglycerate mutase enzyme, so is converted into 2 phosphoglycerate. So the dehydration takes place. So this phosphoglycerate dehydrates to form phosphoenol pyruvate. So this phosphoenol pyruvate is a high energy molecule. It transfers one of its phosphoryl group to the ADP. So that ADP is converted into ATP, and this phosphoenol pyruvate, after transferring its phosphoryl group to the ADP, is converted into pyruvate. So this is the EMP pathway for the conversion of glucose into pyruvate and ATP.